I am Dr. Skala Kumari, Assistant Professor, Department of Botany, Holy Cross College, Autonomous, Nagarkoi. I am going to share about somatic hybridization.
We want to see the topic somatic hybridization. What is meant by somatic hybridization? Development of hybrid plants through the fusion of two protoplast or two different plant species or variety which produce a new plant that is called as the somatic hybridization. How the somatic hybridization we have to isolate? First of all, isolate the protoplast. Next, the fusion of the desired protoplast between the species or variety. Next, the identification and selection of a specific somatic hybrid cell. After the specific hybrid somatic cell, we want to culture the hybrid cell from the culturing regeneration of hybrid plants. Okay, let us see about what is meant by protoplast. Protoplast is known as the naked plant cell, refers to all the components of plant cell excluding the cell wall. Protoplast is the biologically active and most significant material of cells. The plant having the cell wall is a physical barrier. It composes of a mixture of cellulose, hemicellulose, pectin, lignin, lipids and proteins. What is the history? Hanstein first introduced the term protoplast. The Hawking first isolate the protoplast by enzymatic method. How we have to isolate? Two steps. One is mechanical method, next one is enzymatic method. Mechanical method means from the plant tissue, the cell is plasmalized. After that, cutting into small pieces, the leaves are cut into small pieces. By the mechanical shearing, we have to release the protoplast. From the protoplast, we have to select the particular viability protein. Next, enzymatic method. After the leaf sterilization, remove the epidermis. After removing the epidermis, we have to apply the enzyme by one-step method or two-step method. One-step method means first pectinase, next cellulase. Two-step method means a mixture of pectinase and cellulase. Together we have to put it inside the medium. We have to identify the protoplast. Next, fusion of protoplast. Two methods of fusion. One is spontaneous fusion. Next one is induced fusion. The spontaneous fusion, two types, intrageneric and intraspecific. The intraspecific means the protoplast fusion is the cross between the same species and an individual which involves the strains of the non-isogenic one. Next, intraspecific fusion is the crosses between two different species. Some of the examples are given below. Next, the protocol. First, we have to select the young plant. Next, leaf sterilization, epidermal peeling. By the enzymatic or mechanical method, we have to get the protoplast. The protoplast is undergoing plating and culturing. We get a new hybrid plants. Next, viability test. We have to select the viability test. Different test, FDA, phenyl-safranin, chalcofluor, white stain. This three staining, the FDA test, moreover the phenyl-safranin staining test as well as the CFW staining test to detect the viability of the protoplast. Culture of the hybrid cells. After getting the viability protoplast, we are going to hybridize the cells with the help of media. The protoplast regenerate a new cell wall and new cells and forming a callus. With the help of the callus, the medium lacking mannitol and auxin, the embryo developing into a new seedling and finally it mature the plants. Regeneration of hybrid plants. This plants we call it as the hybridization plant or somatic hybridization. Advantages. By the novel method, we have to produce the pomatro. Next, fertile diploids, transfer gene for disease resistant, heterozygous lies in a single species, production of unique hybrids of nucleus and cytoplasm. Thank you.